Hi, everyone. Hope your Monday's going well. Thank you for joining us at our second, more recent Neurotherapeutics work group from the GCC. I'm John Allen, sort of chairing the session today. Good to see you all virtually. Uh, I don't know about you all. I was vaccinated not too long ago. As is my wife. My wife works in a COVID clinic, and she's been on the front lines giving vaccines. It's pretty stunning. I think they had 30,000 vaccinations in about 8 weeks over the Houston Methodist. So the vaccinations are just incredible. We have one of these beyond virtual sometime in the upcoming year, we're hoping. But thanks for joining us. Today is sort of an overview. We've got various things planned to discuss today, mainly to discuss some RFAs. And then also we've got some science to share a little bit from Guru, who's a guest scientist actually outside of the GCC, is doing some really innovative work with exosomes and pretty neat science related to exosomes that he'll share a little bit about. It might spurn some interest in very innovative ways of thinking about target space and thinking about unique ways of thinking about neurotherapeutics related perhaps to that science. So that'll be fun to talk about. I'm going to pop up my screen real quick and kind of give an overview of what I'm planning today. And by the way, this is meant to be a work group, so it's totally informal, right? Even though I've got some slides, it's meant for us to get together and have conversations, ideas, open it up to even just things that are far outside of what our agenda is today. If any of you need help or guidance or thoughts or something keeping up late at night thinking about neurotherapeutics, we can talk about basically anything today and in our groups going forward. I'm going to pop up my slides here. Can you all see that?
Yep.
Okay, terrific, thanks. Let me go into work mode. So yeah, you may all recall we had our first sort of hit-off meeting with our mini symposium several months ago. And this is our first try at sort of having an updated meeting. I scheduled just a little bit of time at the end. I would love to get folks feedback about how often we might have these types of meetings and about topics and things. For those of you that haven't been here or might be new, our work group, the overarching goal of this is to bring together basic and translational neuroscience researchers and collectively get together in this work group to help us advance concepts, potential therapeutics for psychiatric neurological disorders. We'll hear about some RFAs today, but really it's to bring us together and catalyze research interactions, promote new research, solve problems, bring us together to get our thoughts around this important area of, I'd say, translational science, neurotherapeutics. And today, one of the work group goals that we had put out early on was to discuss new funding opportunities. And there's some interesting RFAs, some of them new, actually, and then some of them reposting that I'll go over a few of these today. And maybe it'll, light the fire of interest in some of us or find ways to collaborate with one another by discussing this today. And, maybe new collaborations will come. So just a little bit of background there. Here's our agenda. So till about 1:20, I'm going to spend the next 15 minutes or so just kind of going through 4 of these RFAs that came out recently that are very neurotherapeutic focused and relevant. Dr. Lieza might have some thoughts. For example, I know she and Dr. Green, I didn't see Dr. Green here, Tom, but she has one of these actually in RO3 for one of these RFAs and may give us some insights on that. But there's some of them are very large multi-year grants up to 5 years, very large budgets, to small sort of pilot grants, some in between here, and even some SBIR funds. I'll be going through this and we can just have any discussions along the way, or we can just have a little bit of time to open up to discussion here, through about the half hour, 35 minutes. And then we have the honor of having Dr. Guru Pendyala. Is that how I pronounce your name? Pendyala. Okay, sorry. Thank you for getting me on track here. Dr. Pendyala will give us an overview about his science. About 15 minutes or so for that, maybe 20 minutes. And then lastly, I'd like to discuss with you all, where we're going in the future. What are our plans? How do we envision, timing and frequency of our meetings and perhaps topics that we'll explore in future meetings to keep us all hopefully engaged and learning and enjoying this work group. So there's our agenda. So I've got 4 RFAs here. We'll spend a little bit of time on each of them. Now, caveat emptor, I haven't read all the nitty gritty of every one of these RFAs, but these were ones that I had found, and Dr. Lieza was kind enough to send a few over to me that are definitely neurotherapeutic focused and worth putting on your radar if you're interested in the science and want to find perhaps new funds or create collaborations. So here's one. This is the first one I'll talk about. It's called high throughput discovery and validation of novel signal transducers or small molecules that modulate opioid or other substance disorder relevant pathways. This was a call, an FOA that came out from NIDA, National Institute on Drug Abuse, quite recently, just about I think 8 weeks ago or so, maybe 6 weeks. I put the link here. We will have these posted. If I'm not mistaken, Dawn can help us with that, or you can email me directly anytime. I can share the slide deck for you for your links and things. This is an intended RFA really to utilize high throughput platforms. They could be either for classic drug discovery, like we have a target, maybe it's a novel target that could be relevant for modulating opioid relevant pathways, or even other relevant biochemical, neurochemical pathways in brain for substance use disorders. And they really want to utilize HTS or high throughput type strategies for this. So as it says, the FOA will support HTS type approaches to identify novel receptors, signal transducers, or they could modulate targets for substance use disorder relevant pathways. And this could include dopamine, opioid, cannabinoid, nicotinic, and likely others that are in there if there's a good justification of them being relevant. Applicants can propose secondary screens, validation of high value identified targets, et cetera, et cetera. This is actually a fairly large grant, potentially 3 to 5, up to 5 years of funding support here. I'm going to turn on my pointer. I realize I don't have it on here. Up to 5 years of funding support, up to $400K direct costs. It envisions certainly probably potentially a multi-PI depending on the approach, how it was going to be done. But I envision kind of canonical drug discovery type approaches could be done for this. Sometimes those may not be the most innovative approach. So they've left this fairly ambiguous and open. The important thing for this RFA is they want HTS, scalable type science to be able to approach it. Possible approaches would be, high throughput screening at a target, or potentially like a phenotypic screen for a pathway that's relevant for opioid use disorder or psychostimulant disorder, or relevant pathways therein that are known to be responsive to opiates or other drugs of abuse, and or genetic screens, even in model organisms. They suggest in here CRISPR type screens. I imagine shRNA type library screens, which would be a very large library for shRNA. You'd have a relevant, maybe scalable assay in a relevant tissue. It could be iPSC derived neurons, perhaps. It could be, primary neurons, perhaps. Could even be a whole animal study, and perhaps zebrafish. They've mentioned these various models. So they intend to commit about $3 million, not a large amount, but fund 2 to 4 of these awards in fiscal year 2022. Application deadline coming up this fall, at least the first ones. And then there are subsequent deadlines that will be coming up after. So this is interesting. Definitely could utilize the type of approaches you'd use for drug discovery or screens, but they leave it fairly open. I think they want to elucidate new biology that may underlie this for signal transducers, right? Signal transducers is awfully broad, but I think also you want to talk to the contact here at the NIH to see if traditional screens, which I envision being able to be used here. For example, Dr. Leeza and Dr. Green and I are interested in some GPCR targets that are, heretofore fairly not studied orphan GPCRs that make up a large percentage of the genome for G protein coupled receptors. There are many very interesting orphan GPCRs exclusively expressed in mesolimbic dopamine system in the nucleus accumbens, and these are interesting targets. Some of them are actually in current drug development interest in pharma as well as at NIDA, others not so much. We're interested in one of these targets called GPR12, the unambiguous and unexciting name G protein receptor 12, because it's an orphan. It doesn't have a neurotransmitter or neuromodulator yet assigned to it, but finding small molecules that can modulate it, using the right validation screens, using HTS approaches for that is one example of how we're thinking about maybe targeting this RFA. So there's that one. If there are any questions, I can answer them now, or we can leave it up open after we go through some of these RFAs if there's questions. But this is one to put on your radar. Here's the next one. This is, many of you no doubt have heard about, maybe even applied to already, for the HEAL initiative, this very large initiative at NIH, primarily through NINDS and NIDA, funding from their budgets for the HEAL initiative. This FOA, is a very large effort with multiple types of funding mechanisms to develop safe, effective, non-addictive therapeutics to treat pain. And ideally, those are going to get away from the opiate-related agonists that, of course, are creating a major problem historically and currently. Lots of overdose deaths, lots of dependency on opiates, great analgesics, but of course that abuse liability. So the NIH has these very large, very complex systems with the HEAL initiative to fund, vast science, sometimes really big science, all the way from IND, investigation of new drug filing at the FDA, all the way through phase 1 type studies. And then hopefully they envision some of those projects getting picked up, with partners in pharmaceutical industry, no doubt. But the R34 is one of these, the U19 one, and then the UG3, They stage these in this HEAL initiative to say, if you don't have real good traction with a team yet, to bring scientists together that may be experts in pain analgesia, experts in drug discovery, HTS, and experts maybe medchem, bring these groups together, set up a screen, be able to do initial screens, validate that you can do that type of work, bring the team together, and get that kind of work. You get 2 years of funding for an R34 type mechanism. If you don't have anything, nothing's really validated yet, but maybe new ideas, new approaches, you need to get screens going. There's pretty hefty budget, about $500K per year for the R34, up to 2 years. Then they envision if you've hit the benchmarks, you'd be able to transition your R34 or apply directly in if you already have this sort of, good foundation of a team, or people have really great validated targets and are already sort of ready to move toward identifying new leads, or perhaps already prosecute a lead, and that's the U19 mechanism. Some of you may be familiar with these. These U19 mechanisms have been set up by NIH to bring together team science. And usually they're 3 R01 type grants. They could even have an administrative core that's in there, potentially being 3.5. You may be able to screen 4 R01s. The U19 grants are very large, up to 1.5 million per year. They undoubtedly are MPI related, and then there'll be a lead PI that's on that, or a contact PI. But essentially they're set up to be MPI type grants. And this focus, at least with the HEAL initiative, would be to already have a lead in hand, or really good probes that are going to be able to really jump into medchem right away. Structure activity relations could be driven to actually make a drug-like molecule that could be put into the relevant analgesic models. The one thing I'll say is there's a whole bunch of different types of pain, as you may already know, neuropathic pain, central pain, peripheral pains, all these different, there's ocular pain, there's all these different types of pain. There's really a vast opportunity here in different forms of pain. Many of them don't have good analgesics, in fact, for subsets of pain. And there's places to go and really, enter new space. And it's great that they're putting this money in. If a project's quite, already in hand lead, you really got a great lead, you already know it's pharmacokinetics are appropriate, you've got in vivo studies already validating it, et cetera, and it really has been a chosen lead, perhaps for the next stages of drug development, then the UG3 is an appropriate mechanism. And that could be a very large budget. And the idea coming out of that is to take the money and the project, multi-team efforts, probably a clinician on board as well, and bring things into investigation of new drug filing at the FDA and bring them into phase 1 potentially. Clinical trials are optional for the UG3. There are no clinical studies for the prior awards. So, 5 years and up, and then these are rolling, ongoing awards, definitely neurotherapeutic focused, around making a safe, non-addictive, effective pain analgesic for the future.
I have a question, John. Do you know the difference at the administrative level between U19 and U01? Or maybe someone of the GCC might answer this question. I mean, is it a matter, just the mechanism-wise, grant mechanism-wise?
Yeah, unfortunately, I'm not familiar with that one, Fernanda. I don't know if anybody else has had U01 type grants.
Yeah, there is a call that I've seen that from NIMH. I didn't bring it for today discussion, but it's a U01. So I wonder, if anybody, maybe Dawn or Shannon, has any knowledge of what the differences are. They're both contract, NIH contract, but I wonder if you have any idea of what the difference is.
I can quickly chime in because I've been part of this UO1. One of them we got a score, but one didn't fly. But
Okay.
technically, for what in the UO1, like you mentioned, is that it's kind of a contract with it's multi institutions where we work along. So you bring in your expertise in in something in X, and I bring in something from Y, and then let's say Dr. Allen can bring in something with the Z component. So you kind of put this out nicely, and the budgets range anywhere. Sometimes these UO1s are generally about a size of an RO1, modular RO1, about 250 to roughly 250. But depending upon the scope of what they want, if it is like specifically to high throughput screening or things like single cell sequencing and stuff like that, the price is very expensive. So there you can go up to more than $1 million, million and a half, or 2 million, for example. So I have colleagues who recently got an UO1 for the Scorch mechanism as part of the NIDA at Yale. So Dr. Serena Spudic, whom I know her very well, so she got a $15 million grant over a period of 5 years. That is the total amount. So assuming
And that is a UO1.
it's a UO1, yes.
Mm-hmm.
So yeah, it depends upon the scope, how the UO1s are generally cut out. Most of them are discovery-based things, and for sure it is technically, they want a multi-site to bring in their experiences together, expertise together.
Thank you.
Yep.
Great, thanks so much for that.
I had also a question about this one, this HEAL, because it says certain non-addictive analgesics. So the question I have is, when they look at this, are they really looking for things that replace the opioid and the pain, area of opioids, which is very different than other pain areas that you mentioned. So I didn't know, if they're looking at neuropathic pain, I mean, they generally don't use opioid for treating neuropathic pain. So I don't know how broad this really is, or in review, are they really looking, is this really going to replace addictive medications like opioids, as opposed to just treating pain, which is like, phantom limb pain or whatever, all these varieties, visceral pain, which opioids don't really treat very well. So I don't know if anyone has any thoughts or have looked like at ones they've awarded to see if that's filters through in the reviews.
Yeah, I think just a key thing is they don't want any addiction liability at all. And so in these, if you're like at the U18 or U19 mechanism or beyond, you have to have a clear strategy with which you're going to, strongly, robustly demonstrate there's no addiction liability, there's no dependence reinforcement type models, that you'd see with, substances of abuse commonly. And that's a big thing. And that's, and the idea, yes, I mean, they would like to replace opiate analgesics. If you could have a good, nociceptor regulating spinal cord analgesia mechanism, would be not the mu agonism, but something else, that would be attractive to them. But, that's a really great, NSAIDs, right? Going to Greg's question, we don't need to replace NSAID type pain, right? We don't need to treat headache, migraine's a form of pain, how far would they go with that? But I do know that that's a key thing, that they don't want any addiction liability. And that's a big part of it going forward.
So I just want to build more on what you said, John, is, you want to probably have a chemist on board for these kind of things. So like Greg was mentioning, okay, you don't want to give morphine. Morphine, after a while, it gets addictive, or so is oxycodone or any other opioids, prescription opioids. So you want to kind of alter the structure, make that new compound, whatever it is, that you modify. And then you want to make sure that it alleviates your pain, and then it is also non-addicting. So they basically, looking at what the way it is they want you to find the holy grail here, for addictions, which will not make a person feel addicted to taking more and more, but at the same time can also alleviate any problem. So that's the kind of thing that they are looking at. And there was a U19 recently, or U18, sorry, that was out last year. And they funded up to like 50 grants, I believe. It was one of the program officers, Arud Ramchandran, So I think Fed, we had a meeting with him, I think, sometime back.
Yeah, actually, four out of 50 of these awards are actually in our department.
Yeah.
So Dr. Allen is another recipient.
There you go.
Yes.
So I'm preaching to the choir here. So.
Oh, no, not me. Actually, Dr. Cunningham in our department and a couple of my colleagues are, but I'm not fortuitous enough to have gotten one of these
Okay.
yet.
Oh, I thought
But
you
yeah.
were. Okay.
No,
Oh,
no, not yet.
sorry.
No. My colleagues, close collaborators on other projects, but I'm not that one.
Yeah,
But-
we just missed it by a whisker. We just missed it by a whisker. So.
Oh, okay. Well, perhaps in the near future. So many of you know about these already. So I just thought I'd put it out there. What I do like about it is there's many stages you can come in: early, mid, and even late. If there's something really compelling that's already almost ready for IND, and come in here at different places and keep things going. And I also like that, they've got continued funding for many years here, so that they envision, if you go from an early stage like the R34, then you get to these benchmarks and accomplishments, and you go to the U19 type mechanism forward. And that's pretty neat. And it's great to see that they're trying to do this. So here's some more. I thought I'd just continue here to keep on time. We've got, you probably heard of this one. I know that Dr. Leeza and Dr. Green actually have one of these. This is just reissued. It's going to be deadline July 15th. It's a pilot project. It's a small RO3 type grant, but it's a compendium grant that's supported from the Illuminating the Druggable Genome, or the IDG effort at NCATS. And if you're not familiar with that, that's a sort of broad, internal funded, NIH funded effort from multiple sites that are working on ion channels, GPCRs, protein kinases, sort of like the three major druggable gene families, and elucidating underlying biology and new target space in those three druggable gene families. And they have these pilot grants that come out that they want to promote other investigators to do science that's related to any of that target space. It could be hit the lead. You could do medchem, you could do screens, you could do underlying exploratory biology to elucidate potentially one of these understudied proteins. Like the GPCRs, there's a large number of the orphan receptors I mentioned a little earlier. There's a whole list of them, many, I think 100 of them or something, 80 something on that list. Most of them ill-defined biology, physiology. So classic things like, shRNA or knockout studies to understand phenotype. Those are approaches to do that. And maybe Dr. Leeza, I don't know if you have any thoughts about your experience with the RO3 and.
Yeah, I can say so. So we, Dr. Green and I, it was an MPI, but Green is the corresponding PI. Had an RO3 for GPR. What was it? GPR 12. Right. Yes, I always get confused of all of these numbers. So it was pretty much structured on elucidating the biology of it and its of the target and its relevance for a basically substance abuse disorder. And so those are small grants, but I think they're pretty nice. Actually, they were initially. So the grant was initially routed or assigned to NCATS, but then, it was moved to NIDA. It was then recoded in a way and funded by NIDA. So one thing I want to mention, there are two targets that are currently there are multiple targets, as John said, Dr. Allen said. If online there is a link you can click and see how many there are. There are two particular ones that are really in line with what we do in our lab. And so there is one of my senior postdoc, he's a research scientist, who is actually planning on to applying for these small RO3, particularly for a kinase pathway for which it's totally unexplored, and it has, we have some very interesting data for it. So we'll, he couldn't, I mean, the senior scientist couldn't make it today, but I'm just speaking on his behalf. So. And I believe last year, we had a meeting, one of our first, I think it was our first mini symposium for the neurotherapeutics, or maybe it wasn't, it was just a meeting, but we had a meeting with U of H, College of Pharmacy. Greg was there, Dr. Cooney was there, and it's some other faculty from that department. And I know there were individuals interested in that. I don't know, Dr. Cooney, if you remember that meeting, and I don't know what happened. I don't know if they are still pursuing that.
Yeah, I'm not sure. I haven't heard if anyone's submitted for this one.
Thank you.
Well, hopefully there'll be a repeat of success on this kinase target you're working on, Fernanda. That's exciting. You know, there's a lot of, I've actually applied for this thing twice because it had sort of a GPCR focus, which is mainly what we work on. And interestingly, the first time I applied was sort of doing CRISPR type things to understand signal transduction of several orphan GPCRs in the striatum of the brain. And it got scored. It was sort of lukewarm. You know, it was okay. It wasn't going to get funded though. And then the second one I put in actually was much more target based and does shRNA and things. And what I found from my experience with those reviews is they want basically uncharted territory. And if you're working on any of these sort of illuminating the druggable genome targets, potentially they really like completely unknown. You know, if there's no small molecule, if there's no agonist antagonist, if maybe even there's not even a transmitter or modulator, they like elucidating that biology and bringing out of the dark to understand more, particularly if there's any sort of, you know, genetic association with human disease, et cetera, et cetera. So I don't know, there's a lot of things on the list that actually are kind of well studied and to some extent, but those aren't going to be the ones that are most innovative from my experience from the reviews.
Next he usually does.
Okay.
Yes, or across my feet.
I'm sorry. Okay, if we're all still here, I just
I'm hosting a
hello.
I think that's background noise for somebody.
Oh, okay. Yeah, maybe shut off our mics if we're not talking at the moment. But here's, thanks, Greg. All right, hopefully you can all hear me. I'll keep rolling. Here's the last one. This is called Novel Tools for Investigating Brain-Derived GPCRs in Mental Health Research. This one just came out just very recently, a few weeks ago. Link is there. It's supported through National Institute of Mental Health. This is an SBI opportunity. I don't know if anyone's got active interest in GPCR targets or is doing anything around GPCR targets, but if you've got a small business on the side or interest in developing a business, this is purely technology development though. I've sat on some SBIR type grants for screening and assay development and things and different RFAs. And they really like the idea of small companies developing technology for, you know, next generation platforms for doing small molecule drug discovery for things like, you know, classic targets like GPCRs, or new ways of reading out GPCR signaling that would be amenable to high throughput screens to actually be able to find, you know, through chemical screens to find modulators as you know, initial leads for medchem, that kind of thing. So they put this RFA out. I thought I'd just mention it in case anybody has an interest or does happen to have an SBIR and any of this crosses over with your interest. You know, the classic sort of phase 1, phase 2, 2-year or 3-year grants. The phase 2 are much larger awards. The phase 1 is not a large award, but enough to get things off the ground. So this is interesting, but it's certainly neurotherapeutic focused and a special call there and hadn't been out there before from NIMH. I don't know anyone working on GPCRs with a small business or wants to make a business. I've put my head around this one a little bit, thinking about some maybe innovative fluorescence-based technologies that we're interested with probes that might be amenable to doing some technology, but we're in the discussions phase right now, thinking about it, although we have no spin-off company out of my lab currently, so there's a whole bunch of things that would be needed to jump through before that. So, all right, well, these are, you know, from small pilot grants up to very large grants like the HEAL Award, U19 or the UG3, UH3, many things here. And I think we probably not, you know, covered all territory yet, but there we are. And our group is really uniquely poised to think about these RFAs and find collaborations also. You know, we've got Greg here, you know, fantastic med chemist, you know, physiologist, Dr. Lieza, various folks here with different backgrounds. And if any of these sound good, maybe it'll be an opportunity to open up a conversation with one another and discuss perhaps finding teams to put together for new RFAs here. All right, I'm going to stop sharing. And, you know, we have a few minutes here if there are any further questions we want to bring up or discuss, or platforms that might be of interest here to discuss. If there are any questions, thoughts.
Yeah, I guess a general question to the audience. Have you seen any of these RFA before, or are they all new to you?
Was a little familiar with the HEAL and the RO3 only because you mentioned it to me. So I just from your discussions, I knew about the RO3 and I knew about the HEAL, but I haven't seen. I need to look more in eReporter to see who's actually gotten HEAL awards, to see sort of what they like. I'm not so clear. I have been on one recently. We're waiting for the summary statement to see, you know, what they had to say and the reviews. But yeah, I'm familiar with it, but I've not been successful on it. So we'll see.
Yeah, Dr. Lieza, I think you've applied for, did you apply for a HEAL award at one point with your target space for pain?
Yes, through the company. So
Through the company.
yes, we applied, got a decent score, impact score, 34, and so we'll see what happens if we're gonna get funded or not. So we'll see. If not, we need to work on a plan B.
Okay.
But I mean, we have a plan B in mind. We'll talk to the team members of that grant together, but we do have a plan.
So there are aspects then of the HEAL award that become SBIR type funding, right?
Yes.
I didn't mention those today, but
Yes.
that's actually mentioned. Yeah.
Right. And one important thing is that the HEAL initiative has a mechanism for waiving the budget limit. So for phase 1, you can go up to 700,000 in direct cost. And so there is a list of waiver justifications that the HEAL initiative, and I think it's both NIDA and NINDS and NIGMS also provide. So you can look, if anything falls in that category, then you can claim a higher budget. Like for instance, if you develop a new medication that is under the category that can be that allows the budget to be higher. So there are different types of, but usually again, therapeutics is always allows for a budget waiver. So
Tox alone really will add up. So, well, great. Yeah, I'm thinking about putting in one for possibly June or maybe in the fall, around an interesting target for peripheral pain, a GPCR that's actually an orphan receptor called GPR37, working with Jin Mo and assistant professor over in neuroscience here at UTMB, and also Dr. Jiazhou.
Jun La.
What's that?
Is
Yes,
it
exactly.
Jun La? Yeah. Mm-hmm.
June La, yeah, you know June. Okay, yeah. So we're going to try and put one in. We'll see how the process goes for the HEAL award. We're thinking of U19. Actually, I'm digging into it a little more right now, wondering if we actually have sufficient hit to lead data. If we really should just try the R34 as opposed to the U19. We're in discussions though right now. So anyway, we're actively looking into that.
That's great.
And, yeah. All right. Well, I'm going to stop now. This has been a really great discussion. I hope it's been interesting and useful for you to learn about this, and hopefully not redundant. I'm going to hand over the reins here now to a guru who's going to share some science with us.
Screen share. Do you see my slides? Can you?
Looks great.
Thank you. So, Samya, basically, my research is focused on two arms. One is on neurodevelopment, and then the other one is from a chronic drug addiction perspective in an adult scenario. But then, over the last 1 year or so, I've been kind of heavily invested in understanding prescription opioid use and how it impacts, neurodevelopment, and then importantly, how this further perpetuates as an intergenerational problem. So briefly about me, I'm an associate professor here in the Department of Anesthesiology at UNMC. I am one of the founding members of a new institute called Child Health Research Institute, which is partnered with Children's Hospital Omaha. And then I am also a fellow from the National Strategic Research Institute, which is a UNMC's Department of DOD's affiliate, and we are one of the 14 sites in the country where we are very heavily funded through the Department of Defense. So coming to my lab research interest, like I said, I have several buckets currently ongoing. We basically, I've been heavily invested looking at pre-postnatal opioid exposure and its impact on brain development. We look at drug relapse and sex differences. That's where some of my work is funded by NIDA, so is here as well. I also have collaborations outside of Nebraska on the East Coast, where we are looking at technology development, biomarker discoveries, including developing newer models for brain disorders such as cerebral organoids. And this is something that I really would love to probe a little bit more. And I think this is a great opportunity, this forum with you folks at the GCCs, to look at drug targets for OUDs. And in addition, I've given my training, my postdoctoral training in HIV biology. I've kind of also motoring down, but on a very low pace at this point in infectious diseases such as neuroaids. We have some works going on with Zika and COVID as well. So what is the common thing here? Majorly, my lab is interested in understanding synaptic alterations. We also look at EVs, which now have garnered a lot of interest in given their role as biomarkers, including modulating a lot of these pathophysiologies in the body. And our emphasis is more from understanding it in the central nervous system. We also look at behavioral deficits, sex differences, and like I said, infectious diseases, given the fact that a lot of these drug addicts who do needle sharing are prone for contracting infectious diseases such as HIV, for example. So these are some of our papers that we have published very recently. These are about a year old or so. And this spans, like I said, both neurodevelopment as well as a chronic drug addiction scenario. So we have published using in utero postnatal oxycodone. We have done with nicotine self-administration. Like I said, this is partnering with Hako, one of my collaborators at MGH. We have performed a thorough investigation of these nano-sized EVs as potential markers for both central and peripheral organs. We have written a nice review article about their role in HIV-related neuropathologies. And then, some of the recent works here are a bit holistic systems approach in understanding how pre and postnatal oxycodone exposure impacts development and behavior. And then this is the last segment that I wanted to show, which is the basis for today's little presentation that I have, is characterizing the intergeneration impact of in utero and postnatal oxycodone exposure. So addiction, as we all know, is a brain disease, and I don't have to preach here to you guys. You all know very well what addiction is and the risk factors such as biology, environment, and how they contribute to this disease manifestation. And then, is addiction a family disease? Unfortunately, it is, because there are intergenerational transmissions associated with substance use disorders. And as the adage goes, drunkards beget drunkards. There are also other environmental factors in addition to the genetics, where there is a hostile home atmosphere, domestic violence, for example, sexual abuse, that can contribute to these addiction to propagate in future generations. So opioids and women health needs no introduction, but unfortunately in the last 15 years, there has been a significant increase in, with almost threefold increase in females, depending on opioids. And then especially from pregnant women who, there is a 400% increase, unfortunately, with opioid use disorder during pregnancy. And every 15 minutes, there is a baby born with neonatal abstinence syndrome. So among the most prescribed or favored opioids, prescription opioids included, is oxycodone gets the first rank here. And then what our goal was to see, are there intergenerational effects in offspring exposed to prescription opioids during and after pregnancy? So this is an animal model which is, we published very well, well established in our lab. We do a postnatal exposure, and we also do a in-utero. So this is something that very closely corroborates, pregnant women taking in utero, mimicking drug use during pregnancy. But then this postnatal is also clinically relevant because it mimics two conditions. One is a lot of moms who undergo C-section, they are in pain, and then they take these drugs, especially oxycodone pills, and they become addicted without realizing that these prescription opioids can easily pass through the breast milk and can impact the newborn. And then it also mimics a scenario where there are kids who are just born and they have issues like, for example, a defective heart or a defective kidney, and they need to be, undergo surgical intervention. And then to quell their pain, they are given adult doses, and I see this firsthand here at the NICU, for example, at children's, where these tiny little babies, unfortunately, are given high doses of these prescription opioids. So what we did was we have developed this model, and then we took the F1 pups born, and then they did not receive any drugs or anything. And finally, they gave birth to the second generation. And then what we did was we collected pups from both F1 and F2 at different time points here. For our behavioral, for our molecular analyses, and then from understanding the behavior during early adulthood. So these are some of the stages. My lab, since I said, is a synaptic biology laboratory. We were more interested in looking at changes in synaptogenesis. And then this is some very small pieces of data that I want to show you here, is that we see that changes like body weight, body length, they unfortunately not just they propagate into the second generation as well, especially with the body weight. You see the F2 generation kids, especially the in utero oxy-exposed animals, in the second generation, they are much bulkier and heavier. And we also say changes with the head size circumferences, not much significant, but still, especially at birth, for example, these F1 generations, the in utero oxy-exposed animals, they have a smaller head size, and that unfortunately propagates even into the second generation as well. So we've looked also, given the fact that these are bulkier, or do we see changes in body mass index and Lee obesity? Yes, we see here right at birth, you see that these animals, especially the second generation folks, they have a higher body mass index and also the higher obesity index as well. We performed some RNA-seq on these, especially at P14, because this is a stage that mimics a high level of synaptogenesis. And what we did was we took this nucleus accumbens, which, as you know, is associated with the reward pathway, and we identified some very distinct genes, both in F1, F2, and in common. And then this is just, it's a busy slide, but what it actually shows you is the Klugo analysis, meaning the different pathways which are impacted in these two generations, And long story short, what we really stood out was the HCRT, or the hypocretins or orexins as we call them. And what really stood out was the HCRT pathway, which was significantly elevated in the F1 generation and also in the F2 as well. So what we did was we took different genes, both precursors as well as the respective receptors for HCTR. And what we saw was especially the HCTR1 or orexin 1 is significantly elevated in the in utero and not in the postnatal in both the generations. So why exactly are hypocretins? Most of you know they regulate a lot of these pathophysiologies. They play a role in modulating neurodegeneration, in feeding, in memory, and also importantly in the reward pathway via the interaction with the nucleus accumbens. And as I mentioned in my previous slide, we did find this particular orexins to be significantly elevated in the nucleus accumbens in our animals. So we further went ahead and performed some behavioral deficits by doing marble bearing, which measures compulsive behavior, stereotypy. And as you see here, not just the postnatal and the in utero, especially in the F1, these changes persist longer and are slightly higher in the in utero, even in the second generation animals as well. So we further did some social behavior as well, social novelty, that is a cage mate versus a stranger, like it could be a brother, for example, or a sister here, and then a stranger. And then in social preference, we have a stranger versus a toy. And there weren't much significant differences which we see, but what we saw was a change in trend in especially towards a higher level in the in utero oxy exposed animals, especially in the second this is the first generation data, but what we see is this particular thing also persists but very significantly in the second generation as well. So this is a long thing, but in the interest of time, I just want to kind of wrap up saying that based on our initial published studies, where we see that there are indeed intergenerational effects that are associated with oxy use during and after pregnancy. There is phenotypic alterations, and importantly, one particular pathway. And I specifically chose this topic today is because, we want to kind of identify some valid hits, given the fact that we are looking at RFAs where they are interested either in a particular pathway or a potential hit. And this is something I just wanted to kind of present to the group here, especially if there is any interest with the hypocretin pathways, given their role in drug reward path, in drug seeking behavior. So, this is my group here. Most of the work here was done by Catherine Odegaard and Vicky Shal and Snake Hu. And some of these are my collaborators. And of course, this is my funding. And I just want to let you know that we have collected the preclinical tissues and blood, plasma, and serum from all our studies that I have shown. So we do also have isolated brain-derived EVs, plasma-derived EVs also done from our oxycodone. And, I have studies going on with methamphetamine, with nicotine, for example, with gabapentin, both in the realm of drug addiction and also in synergy with HIV as well. So there is a lot of tissue. I have like 3 negative ADs already filled up with all these tissues from our different animals. It costed me a bit, but I think it's worth the investment. And so it's just that if you have any clinical collaborators like clinicians around at your place who want to kind of, expand their clinical observations and try to corroborate with our preclinical studies, this is something that we can do. And not just for clinicians, even for you folks, if you have any specific interest, like Fair and I, we just created a memorandum of understanding with a MTA, given our interest with FGF13, and she kind of wants to kind of look at from a substance use disorder perspective, but this is what we are. And this is a building, it's a nice fancy building that we are located in. So this is all what I can just share. And I'm sorry if I've rushed through, but I've tried to keep things on time, but just to give you guys an overview where we stand. So this is just one little bucket I showed you out of the 10 different buckets I currently have.
Thank you so much, Guru. That was just terrific. And it's almost with such short time. Thank you for taking time to overview this.
Absolutely.
I've learned a lot. I had no idea you had so much expertise across all of this, and this, of course, the animal model. It's remarkable. I was intrigued by the orexin connection. Merck developed suvorexant not too long ago. It's an orexin receptor antagonist, both I think orexin 1 and orexin 2.
Right.
But have you ever tested whether any of those antagonists would do anything in
R21 that I submitted? And then we also are currently working. Catherine she's actually defending her thesis this Thursday. So we are busy with that. But then we have another student who would be moving forward this particular story of looking at suvorexin. So what we want to do is hit our, half of the population, like especially the in utero, because it's not in the postnatal oxy animals that we see this effect, the hippocretins. It's only in the in utero exposed animals. So what we want to do is right after birth, we want to take half of the animals, just give them, saline injections, and then half of them will get the suvorexin treatment. And then we will try to see if we are able to reverse, like I showed you, they had higher body weights, higher obesity index. So if you are able to quell those physical attributes to a certain extent. And then importantly, what we are currently doing is trying to hook these F1 and F2 guys up to, just oxycodone self-administration during adulthood, and the big picture here is if we can do a suvorexin therapeutic regimen and then see if you can quell drug-seeking behavior, not just at, later adulthood, but right in the very beginning. If you can change the head size circumference, if you can change the phenotypic attributes, in short, if I can say, then we have something to do. So I think this is the kind of stuff that even an STBR or, like a company might be interested, not just with suvorexin, maybe they have some other drugs out there in the market which needs testing.
For sure. Yeah. Intriguing. And did you see anything with the F1 or F2 with diurnal rhythm or anything with differences? Because, it's incredibly strong. It's keeping me awake right now. Orexin,
You
right?
are trying to steal words from
Sleep,
me.
because of narcolepsy and the link. I mean, did you ever see any differences in wakefulness and how?
No. What we want to do is run some actograms on these. So speaking of that, my PhD is in circadian rhythms. So
remarkable. Okay, well, that's
So
a great connection.
we want to kind of look at we want to look at the suprachiasmatic nuclei where the biological clock is in the brain, for example. And then we want to kind of perform a high throughput screen and see which critical genes associated with the biological clock, like period 1, 2, cryptochromes, Rev ERB alpha, all these different genes which modulate our sleep-wake cycle, whether those are perturbed, and that can be another reason for these animals to be dependent on substance abuse. So that's something I really wish I could find another grad student, but that's something that I really would love to. But if there is a collaborator like you or anybody interested, I'm very happy to move this forward.
Terrific. And just the last plug, I grew up in Lincoln, Nebraska, so it's nice to see someone.
There you go.
Small world. Any other thoughts, anyone, or route gurus, science or questions? It's really neat work. Okay.
Yeah, one thing I was particularly interested in, and that's how I got in contact with Guru and his team. It's the ability of preparing exosomes, and I guess maybe we can, as you said, we can talk about that another time. But I thought that was really
I was
fantastic.
planning to present works on that, but since I didn't know about it, though just that others who don't know, we are husband-wife. So we barely talk science at home. So I didn't know that she already responded to the group. So,
Yeah.
But she will be happy to present about some recent findings, not just from our oxycodone, but importantly from morphine acute and chronic exposure, and also from our HIV TG rat. So there is a new concept that her lab has developed. It's on mitovesicles. So she would be happy to talk about that maybe, in a next meeting. And that is something that, can further expand the scope and maybe even bring on some new collaborations.
Thank you.
Thank you very much. And I guess we just have about a few minutes left. I just wanted to end. First of all, I hope this has been a fun afternoon for you, perhaps over lunch it is. And, we've been discussing about how often we should have our meetings. And, I think monthly is probably too many for this type of work group, but bi-monthly, every 2 months, or even quarterly. Does anyone have a thought? I know everyone has busy schedules. It's a question of, doing this over a lunch period, learning something new over lunch, kind of, a little bit of a work group work with lunch. I think people could fit it in, but is bi-monthly or quarterly doable for everyone? Does that seem not too much?
I could do.
Depends on the content.
I think 2 months is ideal if we can present because given the fact that most of these RFAs are coming up around summer to early fall, if there is a way we can have them even monthly, once one of us can talk and then try to start putting things in perspective, I'm game for that. But again, I would go with whatever the team says.
Sure, sure. Well, it's just sort of a new regathering of the work group that we'd had previously here in the GCC. So I don't, how often did you have meetings before, Fernanda?
Oops, sorry. So you know that the work groups meetings were kind of launched last year. So just to give you a perspective, Guru, so the mental health research cluster includes about 120, 140 members. And so we do have at the moment 6 work groups, but you know, those work groups are intended to be formed and sunset if needed. And so we have been following the instruction or guideline from the GCC, which is pretty much having once a month meeting. So that's what the GCC allow. I mean, I think the GCC availability in support is once a month for us, but we can meet anytime and, you know, outside the GCC platform. We don't have to stick with that if it comes you know, if it's more. So right now, because we have 6 work groups, you know, we kind of rotate them around, but some work groups are less active than others. So I'm pretty sure that it won't be a problem to, you know, to have more than what we planned. So just to summarize all of what I said. So far we had basically 2, I believe, work groups meeting so it's basically 1 every 6 months on average for us. You know, for your group, John.
Right.
But we can
Right.
change that. You know, we can either open new slots if some are already taken, or you know, we can just take more than that what we have done before. It's really on the basis of need and interest. So
Sure. Yeah. I was thinking every couple of months or quarterly, but it depends how we want to envision it. You know, we didn't get a huge group here today. We got some feedback after the mini symposium, and it was actually very positive. Most folks said that they would attend and, you know, were interested in this as a platform to, you know, create collaborations, to learn about RFAs, to, you know, learn about some new science. And just there's various forms we can take. I mean, it could be as simple as a data blitz, or we can have someone give a talk while we're also having our work group meeting. I think this was just terrific, Guru. You're very kind and generous to put this almost data blitz together for us. Thank you. I actually find that that's neat and makes the 1 hour a lot more fun. And you end up, you know, you can't get the big picture, but you can learn something new. And I actually like that. There's also the possibility of things related to neurotherapeutics, which is vast, and having, you know, teaching workshops, if someone would be willing to do that, and sort of integrating it in along with the workshop things that we do.
yeah, I think personally, I think if we could do, you know, by every 2 months, basically, I think that makes sense, because I'm worried that if we wait too long, you know, we lose traction, and especially because these RFA are coming up
Right.
rapidly. I think it's important that we kind of keep a momentum.
Sounds good. Well,
so now it is possible though that, like, if we want to, I guess the GCC will have to tell us, I think Dawn or Shannon, I think the 2 months from now will be June 12th. I mean, or around the middle of June, which I think is that date is taken for one of our trainees talks, basically. You know, we have 3 trainees event: junior faculty, postdoc, and pre-doc. And those will are taking one of each slot, one of our monthly mental health research cluster slot. So we probably need to create a new one, but I think I don't really see any problem. Dawn and Shannon, what do you think? What do you suggest?
So I showed June 14th to be set aside for junior faculty. So the rule is that each consortium, you can have 1 event that we can support a month. If you wanted to schedule something, we could put it on the calendar. If you guys wanted to organize it and get everybody registered and stuff, we could put it on the calendar and help with that. But we can support 1 event per consortium a month.
Yeah, yes, but now if we do on our own, would you be able to distribute it to the GCC network?
Yeah, I believe we can, yeah, send the invitation out.
Okay, so I guess it's gonna be up to Dr. Allen how he wants to do it, but again, my feeling is that every 2 months will be appropriate. At least now, but maybe when, there are less RFA coming through, we can decrease. We don't have to be on a strict schedule.
And I think
Well,
some of
well,
these
well-
RFAs, they have multiple dates, some in July, some in October. So I think we should really go in hard, but so I think, and accordingly plan.
Yeah, I think we can let the sort of RFA deadlines guide it as well. What I'll do is I'll, keep an eye open and, I may have missed some things. I didn't do an expansive search for these types of things. Frankly, there might be time to spend another one of these 45-minute sessions or so on things I didn't find even next month. But let me just clarify, Dawn and Shannon, you said one event a month per consortium. So like one a month for the Mental Health Research Consortium?
Yeah, that's right, John.
Okay, all right, thanks. Yeah, well, we have 6 work groups. I don't know how we'll do it monthly then, unless we do it our own, basically, right? That's how we'd have to do it. Well, I'd like to.
Some work groups are not super active. It depends.
Oh,
So
okay.
don't worry too much about that.
Sure.
Yeah.
Sure. Well, I mean, we can space it, right?
We have
can see
another
what-
group. Yeah, we have
we
another
can
group.
Figure out a way to work.
We have another group
Okay.
that does a weekly event actually, so we are not involved with the weekly, events that they have. We do support their 1 monthly, and that seems to be going very well for them. So just, if y'all want to coordinate on that, I think it'll work fine for you.
Okay, well, that's great to know. There's no barrier then. That's terrific. Okay, I say we try every 2 months, and I appreciate your thoughts on that, Guru. And we can update the RFAs then, and I'll give some thoughts about having some science shared as well, perhaps from one of our members, and reach out to someone that might share some cool new science that's going on related to neurotherapeutics. Well, all right. Thanks everyone for joining us today. Reach out. I'll try and have these slides posted. Maybe Dawn and Shannon can provide it if you want links. And great to see you all. And thank you again, Guru.
Yeah.
Thank you.
Thank you so much.
Have a great day.
We'll catch up.
Thanks,
Thank you.
everybody.
Thanks, everybody.
Bye.
Everyone.
Bye-bye.